And I'm going to be going over some of the answers to this test review. I'm not going to go over all the definitions here. You should be able to find those in your notes. But in particular, make sure you know the difference between codominant and incomplete dominant, and also the difference between multiple allele and polygenic. And then also make sure you know what kind of letters you're supposed to use. Are you using capital, lowercase, the same letters, different letters? Are you doing X chromosomes with a letter on them, that kind of thing. For number 2, a woman who is homozygous for freckles and type AB blood has a child with a man who is heterozygous for type A blood and has no freckles. So I'm just going to do another page here so I have some room. So the woman is homozygous for freckles, so we'll use F for freckles, big F, and she has AB blood, and the man has no freckles, that's recessive, so he must be little f, and he is heterozygous for type A blood, so he must be AO. So then we need to figure out all of the possible combinations of alleles that these could have. And so for the woman, all of her eggs are going to contain a big F allele because she doesn't have any other alleles for freckles. And then half will contain an A allele and half will contain a B allele. So I can fill out this whole square and it would look kind of like this. But you should notice that it's duplicated here, right? There's 2 FA columns and 2 FB columns. So since this is duplicated and this half of the table is going to be exactly the same as that half of the table, I don't actually need these 2 columns to figure out my ratios. Now, if that confused you, just go ahead and draw the whole square, but this is kind of a shortcut. And then for the father, all of his sperms will have a little f allele in them, and half of them will have an A allele and half of them will have an O allele. And so same thing with this, since it's duplicated, I don't need these 2 columns either. So just because of what these genotypes were, the fact that they were homozygous for these traits, I actually can do this with a much smaller square. Again, if that confuses you, just do the whole square. But when I fill it out, so I get 1/4 of each of these genotypes, and for phenotypes, all of them will have freckles, since freckles is dominant, and I will get 1 out of 4, 25% freckles with type AB blood, 1 out of 4, 25% freckles with type B blood, and 2 out of 4, 50%, 1 out of 2 freckles with type A blood, because both of these would have type A blood. Right, next we have the cat fur genetics, and so that is a combination of codominant and sex length. The cat fur colors are on the X chromosome. So the male black cat would be XBY, and the orange female cat would be XOXO. So if we do our square, sorry, that's a pretty ugly square. Looks like that. And so we would get 50% calico females and 50% orange males as a result of that cross. Polygenic traits in humans, some examples include skin color, eye color, there's others, height, anything that has like a lot of variation is probably a polygenic trait, and a lot of traits are polygenic, just meaning that more than one gene is controlling that trait. White cow and red cow produce a calf that is spotted red and white. It contains both red and white, so that's going to be codominant. Spotted cow has a baby with another spotted cow. What's the percent chance they have a white calf? So spotted cow will be RW, and there is a 25% chance they will have a white calf. Linked genes, so this is important. This is where you have genes on the same chromosome that are close together, which means it's unlikely that there's crossing over between them, which means they tend to sort of travel together rather than independently assorting or, randomly ending up in any given gamete. So since they tend to stay together rather than splitting up because there's not a lot of crossing over that's able to happen, that is going to throw off our Punnett squares, and we're going to get not our expected ratios. All right, so thick stems are dominant over thin stems, and curled leaves are dominant over straight leaves, and we have a group of plants that is heterozygous for both. So in a double heterozygous cross, we expect our ratios to come out 9 to 3 to 3 to 1. 9 being the having both dominant traits, the 3s being the combinations of dominant and recessive, or recessive and dominant, and the 1 being having both recessive traits. And so I can do the math in here and figure out, what the ratios of these are. But honestly, I can tell just by looking at it that something is up here. So we have 700 plants with thick stems and thick leaves, which is the dominant combination, which, yeah, we do expect that one to be the most. But then we have the next most is 500 plants with thin stems and straight leaves, which is my recessive combination. And according to my ratios, only 1 out of 16 should be that recessive combination. And it's actually the second most. And then here are my dominant recessive combinations, which should be 3 out of 16. There should definitely be more of these than there are of these, and that is not the case. So these ratios are definitely too far off of this expected ratio. Therefore, I can assume these genes, yes, they do appear to be linked. Now, if you're wondering, how do I know, how far off of the expected ratio is far enough off for me to say that I think the genes are linked, or I think there's something is up with how the Punnett square worked out? And that is a good question. That's something that we will address in a future unit. There is a mathematical statistical test you can do. But for now, the problems you get, it'll be obvious enough. This was clearly way off. There shouldn't be a bunch more of the recessive than the dominant recessive. And so you can just kind of base it off of, if it's clearly just way off, then that must be a linked gene. Right, this problem, in order to have room, we're going to look at this on the next page. So our parents in this cross, we have tongue rolling, which is a dominant recessive trait, and we're told that both parents are heterozygous for tongue rolling. And then we have a sex-linked recessive trait, which is color blindness. So the woman is XX and the man is XY. And then the woman, it says, is colorblind. So she must have 2 copies of the colorblind gene, so I'll use little n's. The man has normal vision, so he must have a big N on his X. And then the Y, again, doesn't get any gene on it. So I'll set up my Punnett square. So for the woman, all of her eggs are going to contain an X little n allele because that's all she has. And then half of them are going to contain a big T and half of them are going to contain a little t. And so I have a similar situation here as I had with the last Punnett square where these two columns are superfluous. Now for the man, half of his sperms are going to contain an X big N and half of them are going to contain a Y, and then half are going to contain a big T and half are going to contain a little t. So I will need all of these columns for him. I could have big T and X big N. I could have big T and Y. I could have little t and X big N, or I could have little t and Y. So in this particular Punnett square, since I don't need these columns, I have each square representing a 1 out of 8 chance. Right, so let's fill this out. Big T, X big N, X little n, big T, little t, X big N, X little n, big T, X little n, Y, big T, little t, little y. Okay, and the problem was asking what are the chances that we have a female child, normal vision, can't roll her tongue. That was the original question. So let's see if any of these are that. Female child, normal vision, can roll her tongue. Female child, normal vision, can roll her tongue. These two are male. Here we have another one that can roll her tongue. Here's a female child with normal vision who can't roll her tongue. And these two are male. So it looks like my odds are 1 out of 8 or 12.5% that they would have this combination. So for pedigrees are, it's a little bit of a puzzle, right? Figuring it out. You need to kind of look at different areas of the pedigree and determine kind of by process of elimination, oh, well, since this part is here, it can't be that. And since this part is here, it can't be that. And then eventually you can get down to what is the correct inheritance pattern. So just to review, autosomal means not on the sex chromosome, so basically not sex-linked. And then sex-linked means is on the sex chromosomes, and so we would expect this to be something on the X chromosome. So first thing I'm going to look at is if I have an autosomal trait, I would expect about the same male to female ratio having the trait. So it shouldn't make a difference whether the individual is male or female with regards of if they have the trait. So I shouldn't see like a lot more males having it, and I shouldn't see like a lot more females having it. And in particular, I'd be on the lookout for that a lot more males having it scenario. And in this case, for sex-linked traits, I would mostly expect to see more males with the trait. So those are some general things to look for. Now, specific patterns. With the autosomal recessive trait, we could potentially see a pattern like this, where we have two parents who do not have the trait and then a child who does have the trait. In an autosomal dominant trait, we would never see this particular pattern because it would not be possible for a child to have the trait unless at least one of their parents had the trait. So we would always see, in a case where the child had the trait, at least one of their parents also has the trait. We would also expect to have no carriers, so no half-shaded individuals if this is a dominant trait, because either they have the trait or they don't. But be cautious with this, because sometimes pedigrees won't show carriers half-trait shaded. It just depends on how they drew the pedigree. But you should know, if you do see the half-shaded carriers, you're not looking at an autosomal dominant trait. And then again, for the autosomal recessive traits, we would see cases where, again, like this, where we have neither parent has the trait, but the child does. And we would also expect to have, again, an approximately even sex ratio of males to females with the traits. And in fact, this situation where the child has the trait and neither parent does, if that child is a female, if we have a female child who has the trait and neither parent does, that also eliminates the possibility that this is a sex-linked recessive trait, because if it was, in order for a daughter to have the trait, the father would have to have it as well, were this a sex-linked recessive trait. She would have to get a copy of the gene from both parents, and the father's X chromosome would have that gene, therefore the father would have it. So this particular pattern right here, with a mother and father who don't have the trait and a daughter that does, really only leaves one option, and that is an autosomal recessive trait. So for our sex-linked traits, again, we expect to see probably more males having the trait than females, and this would be a situation where we could have something like this. Where we have a female carrier, a father who doesn't have the trait, but then a son who does have the trait. And again, we would be looking for much more males having the traits than females, or potentially only males being shown with the trait. And if you have a female with the trait, it would have to be a situation where the father did have the trait. You couldn't have a female who has the trait if the father did not have the trait. Now, a sex-linked dominant trait is unusual. You definitely wouldn't see that as much as a sex-linked recessive trait. But one thing you could look for is a situation where if the father has the trait, then the daughters will always have the trait. So any daughters that come from this father must have the trait, because his X chromosome has the trait and they are getting the X chromosome from him. Now, if the mother had the trait, it would depend on if she was heterozygous or not. If she was homozygous, we would expect that all of her sons would have the trait, and half of her daughters. If she was heterozygous, we would expect that half of her sons would have the trait, and again, half of her daughters. So again, with these pedigrees, you're not as likely to have to draw a pedigree as you are to see a pedigree and have to interpret it. So you can kind of play process of elimination, just say like, okay, let's say if this was an autosomal recessive trait, does this pattern that I'm seeing work? Are there any areas that contradict that where that could not be the case? And if this is sex-linked, does this pattern I'm seeing work? Are there any areas that contradict that and say, no, it couldn't be that? So I kind of do a little process of elimination thing to figure out, again, what kind of trait I'm looking at in this pedigree. And then at the last, just note that there are some vocabulary words that you should know that you might see show up in problems. And also there wasn't a specific question about this, but remember that some phenotypes can be determined or influenced by the environment rather than just the genes. So there could be other factors, again, in determining the phenotype. The example I gave was like human skin color. If you're out in the sun a lot, your skin color will probably be darker than if you weren't in the sun. So that's an example where genes are playing a role, but also your physical environment is playing a role as well.